Hello and welcome to Chapter 3. In this lecture we'll be discussing the laboratory. Whenever we talk about the laboratory there will be several things that we discuss, but we're really going to focus initially on the 5 I's, which include 5 basic techniques like the inoculation, incubation, isolation, inspection, and identification of microorganisms. So, as you can see here, here's some images illustrating some of these things, including inoculation and incubation. We have isolation, inspection, and identification. But really, we're going to go through all these in greater detail as we go, so let's just go ahead and start off with inoculation. The idea behind inoculation is to ultimately, potentially, produce a pure culture. But we should probably define what culture is. Culture is the purposeful growth of microorganisms, also known as cultivation of. Really, any growth that appears in or on a medium after incubation would be considered to be part of the culture. So again, growing microbes on a medium is culturing. So since I used the medium, let's use the word medium. Let's go ahead and discuss medium or media. The medium or media is actually what's going to provide the resources for the microbe to grow. So it's the nutrients, the things that the microbes will feed on. And so the idea here is that in order to culture or grow microorganisms, we need to supply them with the food that they need in the media. So how do we actually do all this culturing? Well, we need to introduce microbes to that media through inoculation. So inoculation is putting the microbes on the media. It's very important that media is actually sterile. So let's define sterile. Sterile is actually something that is free of microbial life. And so if you hear the word sterile, it's meaning devoid of life. Because if we're trying to purposely grow something or culture it, we want to make sure that that's the only thing that's growing. So we must start with sterile media to ensure that. The next I that we should talk about is incubation. Incubation is actually designed to allow us to provide the resources beyond just food resources that the microbes need. There is, again, an optimum temperature for most microbes to grow, and most incubators run between 20 and 45 degrees Celsius. Incubators can also provide gases required for growth, including things like oxygen. So whenever we talk about our incubator in the laboratory, it's actually set at 37 degrees Celsius, which is body temperature. But it also uses atmospheric gases, so it's the same air that you and I use to survive. So what I'm really getting at is the microbes that we work with want to grow inside of you and use the same resources that we do. So there is some potential risk for infection, and I must stress that in the laboratory you need to be safe, because it is important that you prevent that type of infection. Whenever we talk about incubation, this is an attempt to allow for growth, but we haven't really discussed what growth looks like in different types of media. There are several different functional types of media, but 2 of them we'll discuss here are liquid medias and solid medias. Liquid medias are going to allow the microbes, as they reproduce, to be suspended in the liquid, resulting in a cloudiness or turbidity. Solid medias, on the other hand, whenever we have growth, since the bacteria are on the surface of the solid, we'll see piles of bacteria build up in something that we call colonies. So again, in liquid medias, the media becomes cloudy. On solid medias, the bacteria pile up and form colonies. As you can see here, there are differing incubation periods for differing organisms. Some of them like at cooler temperatures, like Listeria monocytogenes or even Mycobacterium leprae. And really the length of time for growth is a variable as well. So do understand different things have different incubation requirements. So we've kind of discussed this idea that, again, we need media to provide the resources. Overall, the medias are trying to duplicate what the microbe would use in a natural setting. And so the idea here is we need to have the compounds that we would find in the normal environment for the microbe. So that means typically we have to artificially or man-make the mixture so that the microbes have everything that they need, since we're trying to duplicate the environment. I do want to point out that some of these organisms will be a little more complex in what they require, and thus it makes growing them in a laboratory much more difficult. So again, we have some general medias that grow a variety of things because they have a whole mixture of different compounds, both organic or inorganic. But again, some things are so specific that we have to tailor medias to them, making sure they have the resources that they need to grow. So, the next thing we should talk about is sterile or aseptic technique. This is very, very important whenever we inoculate media. And sterile technique helps ensure that we're only moving or culturing the bacteria that we want to. Okay? And the idea here that by using sterility, we can ensure nothing exists. So whenever we now introduce something, we can assume that's the only thing that's been introduced. Again, we won't know this until after we've seen growth, at which point you may actually recognize that you have lost sterility and actually cause contamination. Again, this was something that we discussed in the class previously in this understanding of why you need to be here. Again, sterile technique is actually very, very important in the medical field. So there are really different types of media that exist. In fact, we really break them up based off of 3 different properties: their physical state, chemical composition, and functional type or purpose. So let's go in and discuss more of these individually. With liquid medias, these are typically going to be water-based solutions, and they again won't typically solidify above freezing temperatures. And so the idea here is these things in the normal environment stay liquid, allowing for again microbes to reproduce and be suspended in the liquid. We have something that's kind of in between a liquid and a solid, and we call that a semi-solid. Semi-solids actually are going to kind of be this blobbish material. The idea here is they're not fully solid, but they don't stay as liquid either. So again, it's like gelatin that's trying to set up. Okay, and this is actually pretty helpful for things like determining motility. Again, since it's not super solid, that means the microbes can move through it pretty well. Solid medias are going to again be solid, and these are used to again allow for growth of bacteria on the surface, forming colonies. And so these are often used for isolation of things like bacteria and fungi. But really, we haven't discussed what made solid media solid, and to make it simple, it is agar. Agar is basically like gelatin, and it is a complex polysaccharide that comes from gelidium. And the idea is that you suspend this powder in your liquid, and then you boil it, distributing it evenly, and then allow it to cool, and upon cooling, the liquid now becomes solid. And so the idea here is we can turn any liquid media into a solid media by simply adding agar. So to illustrate that idea, you don't know what this is yet, but we have something called nutrient broth. Nutrient broth is just water and nutrients. But we can turn it into a solid media and produce nutrient agar by simply adding agar to that broth. So you can see here the different types of physical forms here. So liquids again are liquids at room temperature. Semi-solids are in between solid and liquid, and then again solid is something that is solid at room temperature. So let's talk about defined or synthetic medias. Again, these medias are designed with a very specific chemical composition. These are again man-made to be used for a very unique purpose. Again, they'll typically contain very pure organic or inorganic compounds that do not vary. The idea here is we're trying to be very precise in what we do. So we're really trying to control the environment, which naturally would be very useful in research. Complex medias are going to be medias that contain multiple contents that are not usually just simple chemicals. Okay, so a good example are things like blood. Blood is actually lots of different compounds put together, right? And so again, complex medias are things that it would be too difficult to actually try to synthetically reproduce. Instead, we find it much easier just to actually use that thing in the media. So you guys have worked with in that first lab blood agar. Blood agar is called blood agar because it straight up has blood in it. Again, it was much easier for us to just add blood than trying to mirror all the different chemicals that we find in blood. So there are other things that can exist, things like meat extracts or even infusions. Milk is actually something microbes do not mind eating. Yeast extract, soybean digest, or even peptone. I do want to point out there can be a pretty wide mixture of things, and we can even add things to complex medias, making them again more acceptable for some microbes that may have particular eating habits. So let's move on and talk about general purpose media for a minute. A general purpose media is intended to grow as many possible bacteria as we can. So I mentioned nutrient agar and nutrient broth before. Those are considered to be general purpose. They have a wide mixture of compounds that most microorganisms would actually be happy feeding on. And as a result, a general purpose media like nutrient agar can grow a variety of bacteria. The idea, though, is that sometimes there's not enough resources for a very particular or fastidious organism to live on. So we must enrich some medias. So enriched medias are going to have complex organic substances that are added to ultimately try to encourage the growth of some things. I do want you to recognize the word fastidious. Fastidious actually kind of means to be a picky eater. They have very specific growth requirements. And so things that are required for growth for these fastidious organisms we actually call growth factors. These are specific vitamins or amino acids that are absolutely necessary for that very specific bacteria to survive. So notice here we're going to discuss kind of some purpose things with selective and differential. With selective and differential, we're talking about two different concepts. A selective media is one that's going to purposely select for a group of bacteria. A differential media is not going to select for a group, but rather show us differences between the groups of bacteria that exist. So if you look at the bottom of here, you'll notice that we're using a mixed sample. That mixed sample is a little bit of everything. So there's a bunch of different bacteria in this tube. If we inoculate it on a general purpose, it's pretty easy to assume that a general purpose would grow almost everything. But whenever we plate that same mixture on a selective media, notice the amount of growth is much less, and now it's only a small subset of what grew before. Again, we selected for what has grown. Whenever we talk about differential, notice we can use the same mixed sample, but whenever we plate it on a differential media, notice we still grow most of what we would have grown before because we're not trying to select for anything here. Instead, though, notice that there has been a physical difference in what has grown. Notice again, colonies have a unique, different appearance that they wouldn't normally have. So the idea behind differential is to allow for some cue that tells us that this is a specific type of bacteria. So, as we just illustrated again, selective medias are encouraging the growth of select microbes. You are selecting for what you want to grow. So this is very helpful in things like isolation, especially whenever you have environmental samples that will often be mixed. Differential media, as we said before, again shows us a difference. We're not just selecting for one thing. We're not trying to control what does grow. Instead, we just want to see a difference between what is growing. So often we use color change as our best way. So now notice down on this bottom corner we have something called chroma agar. Chroma agar is super cool, and that whenever you put a bacteria on the plate, it turns a color. So notice they wrote E. coli in E. coli. They used the bacteria to write the word E. coli, and thus now you can see E. coli appears as kind of a salmon pink color. They wrote Klebsiella with Klebsiella. That appears a real dark blue. Proteus is a gray. Staph saprophyticus is going to be a very light pink. And so the idea here is we could grow a huge amount of different bacteria, but they all appear unique and different. So let's talk about another type of differential media. And you kind of got into this in this first lab, but another type of differential media is actually blood agar. Blood agar can show us differences between types of bacteria based off something called hemolysis or hemolysis. Okay? If you break that word apart, hemo means blood and lysis means to break down. And so the idea here is we're looking for the ability of microbes to break down blood. Some bacteria are better at this and can completely destroy red blood cells. We call those things beta hemolytic. So if you see beta hemolysis, it's the complete breakdown of red blood cells. Alpha hemolysis is still breakdown, but it's only partial breakdown, and what you're left with is a waste product that actually takes a grayish greenish appearance. So alpha hemolysis is partial breakdown or incomplete breakdown. Gamma hemolysis is actually kind of a pretty dumb name because gamma hemolysis is literally no hemolysis. Why we don't just call it no hemolysis, I have no idea. So you can see the different options here. Notice that around the strep pyogenes example here, that all the red of the blood is gone. That now you see that amber color of the agar. So the idea here is this is beta hemolysis. We've completely removed the red of the blood. Notice the patient here also has beta hemolysis. This is not good for this patient. This illustrates that they have a beta hemolytic organism. And to really put this in perspective, if it can break down blood on a plate, it sure as heck can break down blood in you, which as you can imagine doesn't go over well. Notice over here we have this grayish green haze that's left around the growth. This is an example of alpha hemolysis, where we have partial breakdown and a waste product has been left behind. And lastly, we have gamma. Notice a gamma, it's still red like the background because we're not feeding on the blood to survive. So let's talk for a bit about an example of media that's both selective and differential. And the one I wanted to do this with is mannitol salt agar because we won't actually see it in our laboratory, so I'll use it as our example. Mannitol salt agar is made of three primary parts: mannitol, salt, and agar. At this point you should know what agar does, and hopefully right now you're saying it's a solidifying agent, because that's what it does. It solidifies the media, making it solid. And so really there are two parts that are going to help us in our selective and differential features: the mannitol and the salt. Naturally, salt is something that most microbes do not really like. Okay, so we have used it for a very long time through history to preserve food because most microbes don't want to be in high salt environments. So naturally we can use the salt here as our selective feature, only selecting or allowing the growth of organisms that can tolerate salt. So that really means the mannitol has to be our differentiation factor here. And so again, mannitol is something that we can break down and produce a waste product like acid. And so the idea here is if you see growth on a mannitol salt agar plate or an MSA plate, you know that the organism is at least salt tolerant, meaning that it can survive in the salt. But again, if we have the ability to break down mannitol and turn it into an acid, notice that there's a color change here. That color change is the accumulation of acid that is affecting the normally pinkish red phenol red. And so whenever the pH changes, that red turns to yellow. So this is illustrating that this is an acid environment, meaning that we're breaking down that mannitol. Interestingly enough, what this test primarily looks for is staphylococcus. Staph readily grows on your skin, which is usually pretty salty, and as a result can readily grow on this agar. But there are two different types of staph that we're interested here: the Staph aureus and the Staph epi. The big difference between Staph epi and aureus is aureus can ferment the mannitol, making again the yellow appearance, while the epi cannot, resulting in still that pink of the mannitol salt agar. Speaking of phenol red, the carbohydrate fermentation test also uses it. So notice again the default tube here is actually red because again the pH is roughly neutral or normal, 7. And so the idea here though is if pH changes, it can turn to yellow. And again, that yellowing is really indicating a lowering in pH that we call again an acid. So in the fermentation tubes, we're really looking for fermentation that results in the formation of acid. So we're going to break down a sugar and turn it into an acid. So naturally, this is going to be an example of a differential media. this bacteria is clearly different than this one because this guy could ferment the sugar to make the acid. Another thing to point out here is notice that there's a tiny little upside down test tube inside of here. We call this a Durham tube. And the reason it's upside down is so that it can capture gas, because there's another product of fermentation that is gas. And so we can look for the production of acid and gas with this particular test. So the next topic that we really need to discuss is the isolation techniques that exist. The idea with colonies is whenever you see one, traditionally we like to think of it starting from one single cell. So whenever you see colonies that are by themselves, you can think of it as being where one cell reproduced itself a bunch to make just a group now of that same cell. And so the idea here is that we can use techniques like isolation streaks to separate bacteria from one another. But there are some requirements for isolation. First off, we need a solid media. Typically we do this on a petri dish, and again we need sterile media and equipment. So let's illustrate an example here of what's going on with the isolation streak method. So first off, this is a 4-streak method. With the name like 4-streak, you should probably understand that means we're going to streak the plate 4 times. Okay, so let's look at this bottom kind of image down here. Again, this is what one really looks like, but here's kind of a graphic illustrating how we do it. So the first time we have to introduce the bacteria to the plate. So the first streak is going to be that introduction of the bacteria on the plate. Once we're done with that introduction, again, we need to sterilize our loop so that we can now just pull bacteria from the previous streak and continue streaking. Again, since this is a 4-streak, we need to sterilize our loop and make our 3rd streak here. And to finish this out, again, sterilize your loop and make the 4th streak. The idea is pretty simple. That first streak is going to have a huge amount of bacteria in it. But since we sterilize our loop in between, we're going to just drag a small portion of bacteria to the next streak. So basically what I'm doing is diluting down the numbers of bacteria by streaking them out over a firm surface. And so as you can see here, early on this culture is basically a sheet of red, yellow, and gray. But by the time we get to our 4th streak down here, notice that we have isolated gray colonies and yellow and red colonies. This is very helpful because now we can feel comfortable that what's there should be the thing that we are looking for. So in order to produce a pure colony or pure culture, we typically have to do isolation. But from that point on, we can actually do what we call subculturing, where we can pull an isolated colony and put it on new media using isolation streak to prove that that is the only bacteria on that media. And so we would call that, again, at that point, pure. We know it's just that one thing. Another word for this is anexic, meaning free of other living things except for the one that we're actually working with. So you can see here there are differences in appearance in terms of mixed cultures and contaminated. A mixed culture may be purposeful, but notice whenever we see it growing on a solid here that there's roughly equal distribution of the 2 different microbes. So there's roughly the same amount of yellow and gray here. With a contaminant, you can see it is a typically lone or very few isolated little pop-ups that happen in what would normally be considered to be a pure culture. I do want to point out most contaminants that you will encounter will typically come from the air. And so it's very, very, very crucial that while you're working with medias that you keep the media closed as much as you possibly can to eliminate those airborne contaminants. So inspection and identification are the last I's that we're really going to do here. And it's pretty important to understand that these two go together. So there are newer techniques though that are actually a little more effective, safer, and at this point honestly cheaper. So we've kind of moved away from a lot of biochem tests because you are actually actively growing the potential threats right there in your laboratory. So we found typically safer options in things like genotype testing or genetic testing, immunologic testing, or even, again, inoculation of specific lab animals. So I do want to really take a second to emphasize the genetic testing and immunologic testing. These things are really the future. Again, there is variability in biochem testing that we just don't traditionally see in genetic testing or immunologic testing. And so the idea is we can be safer but also more accurate in our identification of the pathogens by using these techniques. So I do want to point out that we traditionally don't just use one of the options. We usually use a variety producing a microbial profile. So you'll look at the physical features of growth, you'll do genetics testing, you'll test it using immunologic practices, and even use micro and macroscopic analysis on things like the petri dish and on a microscope. The American Type Culture Collection, or ATCC, is actually one of the country's largest stores and potentially provider of cultures for much of the research community. It's pretty important that, again, we have continuous cultures of stuff that we know so that we can work with them doing experimentation. But I do want to point out that really anytime that we have levels of microbes higher than we would naturally find in the environment, we now consider them to be a biohazard. So cultures or specimens collected from patients or the environment that can be allowed to grow, those are things that are now a risk, and as such we need to dispose of them appropriately. The most common way of doing so is actually autoclaving or incineration. The idea here is that by using heat we can kill the microbes pretty easily at a pretty low cost. So the next thing we're going to go into is the discussion of microbes and size. And really we're going to, since we're in science, use the metric system. So we'll do our measurements in the meters scale. But again, since we're working on the small end, it'll be millimeters, micrometers, or micrometers, and nanometers. Here at the bottom you can see smallest bacteria is around 200 nanometers, while protozoa tend to be in the low end of millimeters, and viruses will be on the low to mid end of nanometers. So again, protozoa and algae, being eukaryotic, are much bigger than bacteria or even viruses. So you can see again this visual representation. So again, the large things that we would possibly look at being here at the top, like louse and ticks and fleas and mosquitoes, fungi. But as you go further down, we're getting to smaller and smaller things. Again, bacteria like E. coli is around 1 micron in size. And then we get into the nano, which is small bacteria and viruses. And then finally you get down to just components of cells like proteins and DNA. So we need to talk a little bit about magnification before we could really discuss how we visualize and use size of microbes. So with magnification, it's important to understand that magnification results essentially from a complex interaction between visible light waves and the curvation of a lens. And so the idea is by manipulating light, we can actually create an image that is much larger. So in order to fully do this, we should probably talk about refraction for a bit. Again, I said that we would have to, for magnification, bend or change light to create magnification. Well, bending of light is actually known as refraction. So refraction is bending of light, in which will change the angle at which the light is passing through a medium or a lens. And so the idea here is that by manipulating refraction, we can create magnification. So I do want to point out that the image of what you see can be enlarged, and so we need a kind of way to measure the degree of enlargement, and the word that we use here is magnification, or times, or x. So again, kind of our measurement for enlargement is magnification, and we again use units like x or times to describe that. So I want to talk about the kind of development of scopes. Again, the early scopes were very simple, just magnifying lenses. Again, they did a good job of allowing us to see the eukaryotic things and start to kind of see bacteria, but we could no way see viruses at this point. Even with today's compound microscopes, which use 2 magnifying lenses and actually a light source, we still have trouble getting into the realm where we could see viruses again, but it did open up the area that is bacteria. So here's an example of a compound light microscope. Again, this is just a generic compound light microscope. They're all basically the same structure. But you can see here again the different parts, things like the oculars or eyepieces, body, nose piece, the objectives on the objective turret, stage, focus knobs, things like that. I'm not going to spend a lot of time talking about those because again you're going to do this in lab. So let's instead point out again there are two magnifying lenses here. The ocular or eyepiece, that should always be 10, and then the objective lens. Again, there are multiple objectives, but you only look through one at a time based off of which one's clicked into position. Notice again the different sizes include 4, 10, 40, and 100 times objectives, and again these help with magnification. But since there are two lenses, in order to determine total magnification, you need to multiply the ocular times the objective. So let's say the ocular is 10, the objective is 4, so all you have to do is take 10 times 4, which is 40. There are some things that actually affect the level of microscopy that can be achieved, which includes again things like magnification, resolution, and contrast. Again, we talked about this idea that the ocular lens would be again a magnifying lens, that is the closest one to your eye. It does form a virtual image that's received by the eye and then converted into, again, what we perceive. The objective allows us to form a real image, which again is a bunch of physics stuff that we really don't need to stress too much about. But the idea here is, again, there are two different lenses that actually create magnification. And as a result, again, as we already did, we need to determine total magnifying power or total magnification. So it's simply the objective times 10. Okay, so if you're on the 4 times, it's 4 times 10, which is 40. 10 times 10 is 100. 40 times 10 is 400. And then the highest objective we have is 100 times, and thus you would have 1,000 times magnification.
Another important variable in microscopy is resolution and resolving power. It's important to us to make sure that we can distinguish two very separate things as being physically separate, and so the higher the resolution, the greater clarity we actually achieve. The electromagnetic spectrum actually plays a huge role in this. Again, this is basically a spectrum that can have varying wavelengths. As a general idea, the shorter the wavelength, usually the more energy associated with it. So things with very short wavelengths, like this end of the spectrum, have greater potential risk of affecting us. I want to point out that we actually observe stuff here in the middle, which is the visible light spectrum. And so the idea here is stuff that's shorter than visible light could potentially damage our DNA, while things that are longer than visible light are actually considered safe. So here's an example of resolution. You can see a low resolution scope actually does not give us that clarification in terms of what is the cells themselves, while a high resolution scope we can clearly identify different cells. Oil immersion is going to be important in understanding how we can observe things with the utmost magnification and resolution by affecting, really, the refraction. So a drop of oil typically will be placed between the 100 times objective, also known as the oil immersion lens, and this actually keeps a more similar density with the glass, allowing less scattering of light, thus increasing the amount of resolution and ultimately clear at that higher magnification. So you can see exactly what we're talking about here. As light comes up from the bottom, it's going to refract whenever it comes into the glass, but also will refract as it comes out of. Since there is a bigger difference between the air and the glass, we see a pretty big or a very large amount of refraction. If we put oil between the lens and the slide, oil is more similar to the density, and thus we see a decreased amount of refraction. So there is again a refraction index. We're not going to take time to really go over it here. Again, just understand that again light bends as it moves through different mediums that are of different densities. So there are some different variations of light microscopy. 4 major ones that exist, but really only 2 are what we'll care about, are the bright field and dark field light microscopes. But we'll also discuss for just a brief amount of time the fluorescent microscopes and confocal scopes. So you can see here again a bright field microscope and then the dark field microscopy. The difference is really the background and the light that we're collecting in the eye. The idea here in bright field, there's lots of light in the background that can actually kind of decrease clarity along the edges. In dark field, you actually get greater clarity around the edges because you're not collecting the light from the background. Phase contrast, we don't really care much about, nor do we really care much about differential interference. But with fluorescence and confocals, we actually see a situation in which we have cells that are colored, and these are purposely colored. What's interesting about fluorescent and confocal microscopy is we use stains that can be excited and creating basically light. And so these things look like they light up on that background because of, again, the fluorescent or confocal energy activating the electrons of that stain. The next thing I want to point out is the electron microscopes. There are two of them that we really care about. The transmission electron microscope, or TEM. The transmission, again, allows us to see the insides of a cell, or the scanning electron microscope, or SEM, that allows us the surface features. There are scopes that go much lower in terms of magnification, but again, these scopes are difficult to utilize, pretty rare, and as such we won't discuss much about them. It's important to understand that we must prepare specimens for observation on the scope. In doing so, we can again increase our ability to recognize what they are. So it's important for the observer to understand what you're looking for, whether the thing needs to be alive or whether it can be dead, what type of microscope you want to use, those kinds of things. But really for us, we're going to do a lot of what we call smear technique or smear prep. The idea is we're going to spread typically a thin amount of cells on the slide and then allow it to air dry. Once it air dries, we actually have to do something called heat fixing. Heat fixing can be done using a Bunsen burner or an open flame, or in our case we'll use a heating tray. The idea here though is we must kill the bacteria but also stick them to the slide. So heat fixing is an important step that must be done. Negative and positive staining are important topics to discuss. With a positive stain, we're actually going to see a situation where we're going to use a stain that's an opposing charge to the outer surface of the cell. By doing so, the stain or color will actually adhere to the cell itself. Negative stain, on the other hand, does the opposite. Since they are like charges, they actually repel each other. So instead of staining the cell, we stain the background, leaving the blank space as being where the cell occupies. We can see that here. Notice this essentially positive stain here is attracted to the negative features of the cell, causing the cell to become colored. Notice this negative stain, on the other hand, the stain itself is repelled, creating a space where the bacteria actually lies. You can see here that there is an example of a simple stain. Simple stains are what the name implies, simple. It's the addition of one single color just to create a difference in the background and the bacteria. Differential stains, much like differential media, is intended to show you a difference between what is there. So these are often complex staining techniques. One of the more common ones we'll actually discuss here is gram stain. Gram stain was developed a while ago with this idea that we can use different physical features of the cells to show us a different appearance through this staining procedure. So you can see here the gram-positive bacteria appear purple. So all the purple bacteria will be called gram-positive. Gram-negative, on the other hand, will appear pink or negative due to features that are different. Another example of a differential stain includes the acid-fast stain. One problem with gram staining is it relies heavily on something called peptidoglycan, which not all bacteria have. So one cool feature is the acid-fast stain actually allows us to look at another feature in some bacteria that utilize acids like mycolic acids on their outer surface. So in this case, acid-fast bacteria stain pink, and the non-acid-fast appear blue or purple. The endospore stain is intended to show us an endospore. An endospore forms from a vegetative cell. I want to point something out. The word vegetative here is actually in reference to a living active cell. The inactivated cell we would actually refer to as being the endospore. So essentially, an endospore is a protective strategy that happens whenever a cell is in a dangerous situation and it wants to go dormant. There are really two major endospore forming genera, Bacillus and Clostridium. So you can see again the endospore initially develops inside, but the rest of the cell can die, leaving the spore to remain. Specialty stains are also very important, but these again are special and will emphasize certain parts of cells and may not reveal the whole actual process of staining the cell. So here's an example in a capsule stain. Notice we can see the bacteria, we can see the stained background. What is not stained is actually the capsule, which is a protective coating that covers some bacteria. A flagellar stain like this one actually again highlights the flagella so that we can observe these long filaments used for movement.